Hello. Today I'm going to be discussing eyes and their role in vision. So throughout this presentation, I'm going to go through the general structure of the eye, each specific section of the eye. I'm discussing the general function of vision. I'm going to point out two specific medical tests and a single visual impairment that normally affects the eyes. So first is the general structure. So the front of the eye is made up of 5 distinct areas: the sclera, the cornea, the pupil, the iris, and the lens. The sclera is the outside white part of the eye, and it's normally made of collagen and elastic fiber and just holds the rest of the eye together. So light first enters through the cornea, which is the clear, bent front of the eye that helps support focus of the light into the iris and the pupil. So the iris is important for opening and closing the pupil to regulate how much light is passed through. And the lens takes that completely focused light and puts it to the back of the eye. And so the back of the eye is used for mostly reading the light that gets sent there. So the light first hits the retina, which then uses its photoreceptors of the cones and rods to turn them into electrical signals, and these signals get sent through the optic nerve, which leads them to the brain. So now the specific structures. So the cornea, as I already mentioned, is the outermost layer. That is the clear part right in front of our iris. So the cornea's main function is to give 65 to 75% of the focusing power of the eye. And that means 65 to 75% of the light, of the focusing of the light, happens right in the cornea. So it's made of 5 distinct layers. The corneal epithelium acts as a wall from outside materials and holds tons of nerve endings to be extremely sensitive. This helps the epithelium be extremely painful to the touch so that people don't scratch or touch their eyes and know to protect their eyes. Right under that is a very thin Bowman's layer, which is extremely dense and also helps to prevent corneal scratches from penetrating into the stroma, which is the stroma is the, well, it's 90% of the cornea, and its main function is basically the backbone of the cornea. Right? It holds everything together and is extremely flexible and can change its shape. So right under the stroma is Descemet's membrane, which is a thin, strong sheet of tissue that serves as a protective barrier against infection and injuries. It's also comprised of collagen fibers, but different than that of the stroma. The endothelium is the last layer in the cornea. And endothelial cells are essential in keeping the cornea clear, and they allow fluid to leak slowly from inside the eye to the middle corneal layer to keep the water that most of the stroma is built out of in the correct area. The iris and the pupil. So of course in this image, the green part is the iris and the black part is the pupil. So the iris is tasked with regulating the amount of light that is allowed in, and we know that your eyes will get bigger or smaller, at least your iris will open and close. In the case of too much light, the iris will close a little bit. In the case of too little light, the iris will try and open. So the iris is made mostly entirely of connective tissue and smooth muscle fibers, and those muscle fibers are what controls the contraction of the iris. The lens is the part that sits right behind the iris and pupil, and the last focusing part of the eye. The lens is held in place right behind by small tissue fibers throughout the inside of the eye. And the elasticity of the lens allows it to change shape and help change focus depending on where an object you're looking at is. So a further object away or closer it is, the lens can either squish or expand to help see further objects. And the retina is possibly the most important part of the eye, as it's multi-layered membrane that's the back and takes in the light, and is where all the light is focused to. So the inner layers of the retina are made up by mostly bipolar cells, ganglion cells, and the cones and rods. but they also have horizontal cells and amacrine cells. So the cones are mostly found in the macula, almost completely, and the rod cells are found in the outer edges of the retina. So the most important part of the retina is the two photoreceptors, the cones and the rods. The retina is used basically to help transfer the light taken in to the brain, and actually it takes in the signals and turns the light into signals, which are eventually led to the brain. Specifically, we're going to talk about the rods and cones. So the rods, there are 130 million rods in the human eye, and rod cells are extremely important in creating our peripheral vision, and they are very good at reacting to motion. They are used to differentiate light and darkness, and so they're important for changing to night vision and help our eyes adjust when it gets darker or brighter. They are, as I already said before, around the outer edges of the retina and further away from the macula, whereas the cones are very abundant in the macula, and they are the only cells found in the center of the macula, known as the fovea. So we use cones to differentiate colors, and they're also used for more detailed vision. There's much less cones than there are rods. There are only 7 million cones. And there are usually 3 types of cones, for 3 different pigments of color, the red, green, and blue cones, which all work together to help you see the spectrum of color we can see. So specifically, if you wanted to see purple, both the red and blue cones would be stimulated in certain ways and would allow you to see the color purple. So the optic nerve is in the very back of the eye, and it is the actual pathway that the nerves converted from the ganglion cells use to get to the optic chiasma and then to the back of the brain. And so the optic nerves from each eye come to the optic chiasma. And these basically are just the sensory signals from the ganglion cells that eventually create the image in the back of the brain. So the front curvature of the eye causes an upside down image in the retina itself, but then the back of the brain is able to flip that over and create the correct image we see. So the general function of vision. So vision in regards to homeostasis is extremely important for witnessing or noticing danger. Right? An article in Zeiss estimated that we perceive up to 80% of all impressions by sight. So if other senses are less helpful, sight is always the go-to and basically our best way of discerning what something is. So as I already mentioned, the rods are both in charge of our peripheral vision and can notice movement very well. So using that, we can see very clearly if something is coming towards us or movement around us to help us perceive danger and prepare for danger. So I'm now going to discuss 2 medical tests, one that's used to check the acuity of someone's eyesight and the other one to check if someone's colorblind. So the acuity test would be the Snellen test, which is a very common test most people take as kids, and with poor eyesight they may be asked to take throughout their lives. Snellen test is normally conducted by asking a patient to stand a certain distance away and cover one of their eyes and try and read down the list of letters until they can no longer discern which letters are on a row. And then they're asked to cover the other eye and do the same thing, and then they're asked to do the same thing with both eyes open. So the results are given based on which line the person ends up being able to discern. The bottom line in this image is the normal 20/20 human vision. And so what that results mean is if that person can get to that line, then that person can see from 20 feet away what normally a person can see from 20 feet away. Whereas someone with 20/40 vision can only see from 20 feet away what a normal person will be able to see from 40 feet away. And this test is extremely helpful in finding out if someone has either a misshapen cornea or a misshapen eye, and basically if they have nearsightedness or farsightedness. The next test I want to discuss is the pseudo-isochromatic plate testing, otherwise known as colorblind testing. And it's normally used to determine not only if someone is colorblind, but can try to determine which type of colorblindness someone has. So the test is normally conducted by asking a patient to look for specific numbers among the various colored dots. And people with a normal color vision can normally see a number, while people with a deficiency may either not be able to see a number or will see the incorrect number. And well, I mentioned before that this does help determine if someone is colorblind, and they try to determine what type of colorblindness it is. It normally takes many more types of testing to actually determine which type of colorblindness someone might have. So colorblindness is, of course, the either missing of a cone or the faulty cone for a specific pigment, which I discussed either could be the red, green, or blue. So the last thing I want to talk about is the specific vision impairments of myopia versus hyperopia, or nearsightedness versus farsightedness. So myopia or nearsightedness is normally caused by elongation in the eyeball, or if the cornea is too curved, which means that imagine if the eye is being pushed down and is elongated horizontally. So distant objects look blurred, can't see as far. And this can be especially difficult in situations where someone's driving, but it's also possible that they might be able to see closer objects clearer, like they might be able to read a book or watch a TV easier. So about 42% of people from age 12 to 42 had myopia around the world in 2009, and an estimated 30% of the US population has it today. On the other side is hyperopia, farsightedness. And this is caused by the opposite. It's caused by a shortened, smaller eye or a flatter cornea than normal. It actually makes it easier to see further objects, but closer objects are much more blurred, again, like reading a book or watching a TV. It's assumed that an estimated 60% of the US population has some form of hyperopia, but only around 10% have substantial, more hurtful cases. So in a sense, in the version of the Snellen test, someone with hyperopia might actually have a 20/10 vision, where they might be able to see something from 20 feet away, where most people would only be able to see from 10 feet away. But over time, even then, they normally will have to get glasses, and even their short vision might get hurt or injured. Myopia would be someone with a 20/40 vision or a 20/70 vision, where they can't see something from 20 feet away that most people can see from 70 feet away. Both of these are caused by the misshapen eye and the cornea, which means that both are caused by an issue in the eye of focusing the light correctly. So it can't focus the light where it's supposed to focus, directly on the macula. And the rest is just references. Thank you.